All right. Well, we're ready to go. Thanks a lot, everyone, for coming to this talk. I see we have a large audience today, 50 something people. It's my first time hosting this. I offered to help, but I have to say most of the heavy load was actually done by Beth and Elna. So I apologize for some of the mistakes that I make introducing, but it's a huge pleasure to introduce Virginie. I've just written a little bit about Virginie. If you give me a moment, please. So Virginie is currently he's a leading expert in quantitative bioimage analysis, and she's currently serving as a director of the recently created BioVisionCenter at the University of Zurich. While she is also still a visiting group leader at the MBL EBI in Cambridge. She started her graduate work at EFL on life sciences and bioimaging, and also worked momentarily, I believe, at Broad's bioimaging platform in Boston. In 2019, she became a group leader at the MBL EBI, where her group developed algorithms, theoretical frameworks, and practical tools to help transform bioimage data into meaningful biological information. Virginie's research is intersected in mathematics, computer vision, biological imaging, and she's also very interested in democratization of advanced bioimage analysis and promoting open science for microscopy imaging, including development of community standards and next generation data formats, as is the case of OME-Zarr, which is our topic today. On a more personal level, Virginie grew up in the Swiss mountains, I believe, and loves being outdoors, watching birds and nature, climbing and hiking, which she says helps her balance her time at the computer. So great balance, Virginie. She's an excellent speaker and a doodler. She makes beautiful drawings, and I'm sure we will all really enjoy hearing today about the interoperability of bioimage analysis at scale with OME-Zarr. But before that, I would just like to take a chance to advertise a little bit GloBIAS. GloBIAS, well, you can find information on GloBIAS not just on the website, but you can also follow the GloBIAS community on Blue Sky and LinkedIn. You can consider becoming a member, so you can have an active voice on shaping the future of bioimage analysis. Just visit the website and see how to become a member. Another way to being involved is just participating in this seminar series, which is basically a dynamic platform for mostly focused, but not exclusively, for bioimage analysts to allow us to stay up to date with the latest developments. It is designed to cater to intermediate to advanced level of analysts. So if you're just starting, it may be of interest, but it's going to be more difficult to catch up. And it tends to be very focused on practical aspects of high-level content that goes beyond just the basics. The Broad Institute is currently providing help with the infrastructure, which we thank. And just again, a reminder that these seminars are recorded, and some of the questions will be later transferred to image.sc so they can be available to the whole community. So take that into consideration if you intervene or turn on your camera. And just a shout out for the next the talk, which is going to be by Nicholas Condon, be an alert. This is going to be on April. So check out the website for more updates, and please consider joining the community as a member. Here's the link, but you can find it through the GloBIAS website. And now, Virginie, it is really my pleasure to hear you once again and see you once again. The digital floor is yours.
Thank you so much. I'm not sure I can live up to this introduction, but I'll do my best. So I'll share the screen. Hopefully that works. Can someone confirm that you see the slides, please?
Yes.
Amazing. Thank you. Yes, great. Well, I mean, thanks, Gaby, for this intimidating introduction, but very kind. And thanks, Beth and us and everyone else who was involved in organizing this. It's really a great pleasure to be here today speaking at this seminar series. So yeah, I mean, I cannot say much more to this introduction except that I'm Virginie. I'm very glad to meet you all. I saw that there were quite a lot of names that I know in the participants. So great to see you again. If I know you already, great to meet you if I don't know you yet. Today, what I want to do is to tell you about some of the work that we've been doing with OME-Zarr. And this is not necessarily a talk on OME-Zarr per se. I mean, I will do a little bit of this, but this is more about work that we've been doing around this file format. I'm based in Zurich. I was said earlier. One of the things that I do there is to lead efforts at the BioVisionCenter towards developing infrastructure for bioimage analysis, and this is what this talk will be about. I also have another hat, which is that I'm a professor at the university, and I have a research group that does method development for bioimage analysis. This is not going to be what I discussed today, but it's also fun. and these two things go really hand in hand. And we are currently growing the research group in Zurich as I'm transitioning away from MBLBI, where it was before. So yeah, I just wanted to say that as a sort of disclaimer, because yeah, if you're interested more on the sort of research part of method development, maybe get in touch, because we'll have openings in the near future in that space. So without further ado, okay, let's get into this. Okay, I guess this is not really an intro slide that I need for this crowd, but still I have it because that's the nice start of the story. I guess we all more or less painfully experience every day of our life that microscopy imaging generates large amounts of data. And for those of you that have been around for a while, I guess we remember that 10, 15 years ago when we were talking, okay, this is a big data set, this was in the gigabyte range, perhaps. Today, that would be a small data set. A big data set is more like in the terabyte or petabyte range. And yeah, Moore's law tells us that this is not stopping anytime soon, right? So this is great. In my opinion, this is fantastic, because more data means more information, means more need for bioimage analysis and cool methods to develop. So we should all be delighted about this, I guess. And I am delighted about this, but big data also mean big engineering challenges, right? And this is the sort of the part that we don't generally think a lot about on the research side. The fact that coming up with new algorithms is one thing, but making these algorithms run on images that are gigantic is an engineering problem that is not easy to solve, right? And one of the sort of very concrete problems of large data is that they are larger than computer memory. And what that means is that, if we can't load them into memory, we can't look at them, we can't process them, we can't do much with them, right? Besides storing them. And even that may not be straightforward. So how do we go about this? Well, sort of, sorry, intuitively, if you have a very big image, I mean, of course, maybe it is gigantic, maybe it is very heavy and so on, but I mean, an image can always be chopped down into smaller pieces. So we can always say, okay, if I have a very, very big image, what can I do to make it less big? I can just turn it into many, many small images. And great, that will solve some of the problems, like the loading into memory, but not all of them, because now instead of dealing with one image, I'm dealing with many images. And this is sort of the other side of big data. It's not necessarily individual data that are big, it's also lots of individual data points, right? And of course, again, coming up with a method to analyze a single image is one thing. Coming up with a method that smoothly analyzes 160,000 of them is another thing, right? Because when you're in this sort of scale, you need to think about the fact that everything has to be very, very automated and has to be very, very fail-proof, fail-safe, yes, such that you don't waste your time and your compute going through half of this file and then crashing and whatever. So yeah, this comes with challenges. And obviously, one thing that I didn't quite mention is that if you start from a big image, chop it into pieces, these different pieces are related to one another. So perhaps you also want to take that into account in your analysis. This is also not entirely straightforward. So these are not new problems. These are problems that have been around for a bit now. And so, of course, many people have thought about this. Many of the names I saw in the list of participants have thought about this. And one of the things that I think is really beautiful that happened in the microscopy imaging community some years ago is that several people coming from different angles thinking about this problem sort of decided, okay, actually, let's agree to one way to approach that and try and come up with a solution that we all can get behind. And this is what next generation file formats are, this OME-Zarr that I'm going to talk about, that many of you know about. I didn't want to assume that everyone knows everything about it, so I just have a couple of slides on it. At a high level, really, the idea is to say, okay, let's come up with a file format that allows us to handle this sheer scale of data, but that allows us to handle it for various use cases, right? Because of course, having large data is problematic when we want to visualize the data, but it's also problematic when we want to analyze them and when we want to store them. So if we come up with a solution for either of these use cases, we may as well find a solution that solves them all, yeah. And the other thing that I guess we all know and experience is that one of the beautiful things about microscopy imaging is that there are lots of different technologies. There are always new ones that come up. And so similarly, if we come up with a solution to deal with large EM microscopy, volume EM microscopy images, for instance, perhaps we don't want this solution to be limited to EM. We also want it to be amenable to whole slide imaging or large light sheets, live data, or this sort of things, right? And this is really the idea behind this OME-Zarr effort. It's to come up with a solution, a next generation file format that is amenable to large microscopy image data, no matter what microscope we are talking about, and no matter whether we are most interested in data management or visualization or analysis or all of the above, right? I will not go really into the details of what this file format is, but there is this nice kind of three-picture overview of the main features with the beautiful drawings from Henning Falk. The key features of this OME-Zarr format, I would say maybe the most important feature, there is only one that you have to think about for the rest of the talk, is that it is this so-called pyramidal data format. What that means is that unlike classical file format that are written in a monolithic fashion, OME-Zarr allows you to store data in a hierarchical manner that then makes it possible to read and access this data at different levels of resolution and to essentially only access the part of the data that you need, right? And this is important when you do visualization, this is important when you do processing, because again, you don't want to load that whole terabyte or whatever into memory. This is also important when you do data management. Actually, a lot of the OME-Zarr sort of movement started originally with the motivation of being able to store large microscopy imaging data on the cloud. Because of course, if you want to upload data on the cloud and retrieve data on the cloud, you kind of don't want to have to retrieve a huge amount of data in one go, because the likelihood that something goes wrong is just too high, right? So this is one of the big motivation and feature of this, is that it is cloud ready. It is amenable to be used on high performance compute cluster. We'll come back to this. That's one of the things I'm going to talk about. And as I alluded to in the previous slide, this is meant to be very general. This is meant to accommodate equally well data that are 2D, 3D, 3D plus time, 3D plus time plus many channel plus whatever you can think of. At this time, if I'm not mistaken, OME-Zarr is specified for up to 5 dimensions, but it can go beyond, and the intention is that it goes beyond. So yeah, this is really meant to be very general. I mentioned before this pyramidal format that allows you to load only what you need and when you need it. And this is, I think, the best illustration of it that you're going to see in the video here. It's this, sort of like Google Maps style type of image browsing. So that's what it enables here. This is a multi-well plate. You'll see this image in many different forms a lot of time during the presentation. This is a big data set where we're looking at cardiomyocytes differentiating. So you have different wells, different things in the different wells. Each well has many different field of view. This is a multiple terabyte data set. When it is stored in OME-Zarr, what that allows you to do, and here this is in napari, which you certainly all know, is to browse this gigantic larger than memory image smoothly, sort of on demand, because as you zoom into the image, you can also look at segmentation labels here. As you zoom into the image, the sort of appropriate level, the appropriate resolution level of the image is loaded while you're browsing the data, right? And I don't know if the zoom compression allows you to see this, but as the video proceeds, you maybe get a feel for the fact that when you zoom in, you don't immediately have the highest resolution level that the image has at the level that you're looking at, but it sort of gets loaded as you go, right? And this is kind of the power, I think, the best illustration of the power of a pyramidal file format, because that kind of image, as a whole, can simply not be loaded into memory on one go, right? I mean, at least not with the computers that I have access to. I don't know what you guys have access to, but yeah. Okay. So OME-Zarr, as I said, it was sort of, I think, primarily thought about initially for storing data, data management, visualization, and so on and so forth. We have been very, very interested in it, not necessarily for this, but for processing, right? Because, okay, of course, again, the pyramidal file format allows you to access only what you need, and this is great because it allows you to build processing units that operate on very large data at sort of minimal engineering cost. But additionally, and I think this is something that I'm very interested in, that is sort of close to my heart, it gives us a path to think about what it would mean to build interoperable image processing units. And what I mean by that is, again, coming with my method development person hat on, when we come up with a new algorithm, really what we usually focus on is, okay, we want algorithmic novelty, we want to be able to do something that nobody has ever done, right? And so we find a good use case to demonstrate how that works, and we implement it on that use case, and then it's amazing, it beats the state of the art, and we publish a paper. And then you come to me with another data set and you tell me, can I use the same method on that new data set? And I'm probably going to tell you yes, because in theory you can. In practice, it's gonna be a pain because the new data set that you have, unless we are very lucky, probably is not exactly the same type of images that I've been working with. Maybe yours is 3D, mine was 2D. Maybe yours has multiple channels, mine was single channel, this sort of thing. And again, not all algorithms can be easily extended over all of this variability, right? But some can. And when they can, it would actually be great to have a structure that allows us to implement them once in a way that can be adapted depending on the properties of the input. And this is really only, in my opinion, feasible if we have a very constrained structure on how we store image data that is general enough to accommodate all this variability, and that is structured enough to allow me to basically always know, okay, if I have an image data that comes to me, I know how to read it, how to make sense of it, and depending on what I read and what I understand of this file, I know how to apply reasonably my method to it. And it could be that, okay, my method was only 2D, you're giving me a 3D image, so now I know that I should apply it, on 2D slices across the different ways that I can slice my volume, something like that, right? So I think this is the first sort of dream that, OME-Zarr allow us to dream about, is to really think about, okay, can we go towards a world in which we develop truly interoperable image processing unit that adapt to the big variability that there is in microscopy data. And I think if we dream even further, we can think about, okay, if what is well constrained and that gives us a structure is the way that we store the imaging data and their transformed selves after the application of the image processing operation. We can also dream of true interoperability even between programming languages, right? And that would be basically saying, okay, if I come up with a new great method, I implement it in a way that can work on any kind of image data, and that can easily be chained with another method that someone else developed in a different programming language. So we are nowhere near this, in my opinion, but this is what we should aspire to, right? And what I'm going to talk about in the rest of the seminar is essentially your sort of baby steps towards that, right? So there is quite a lot of things happening in the OME-Zarr space, much more than I'll talk about here. If you're interested in this and you're sort of not already part of this nice and fun crowd here, please join us. There is a website here that was heavily reworked and expanded in our last hackathon last year by many great people, so it has tons of information. There is, of course, there are several papers that tell you about the work that has been done in this space. And we have a couple of various community events that I hope are welcoming enough to allow you to just show up and see, okay, is there something that I could do? What is it all about? And how do I sort of join this crew? And the next one is our quarterly community call. This is happening next Tuesday, so not tomorrow, the week after. You will find, again, all of the information on this website. So yeah, please, if you're interested in OME-Zarr and kind of want to go beyond the super fast summary I gave now, look into this. Now, okay, what do we do specifically in Zurich and that I want to tell you about today? So the BioVisionCenter, as I mentioned, is sort of the part of my life in which I think about infrastructure for microscopy image analysis. What we do there is to think about how to contribute to democratize access to advanced bioimage analysis. And what that means is that we want to basically put the latest and greatest algorithms directly in the hands of life scientists who may not have any programming expertise. And we also want to contribute to fair standards for bioimaging data. So as I said, we don't necessarily work in the space of data management per se. We work more in the space of data analysis, but I do believe that there is a lot to do as well to verify things, right? And so for the rest of the talk, what I'm going to do is to tell you a little bit about two pieces of work that are currently ongoing. The first one is tooling development to handle OME-Zarr. And this is essentially a Python library that we've been working on. The second one is a much bigger scope project that is a workflow management platform entirely based on the OME-Zarr format. So let's dive into the first thing. This is ngio. That's a Python library to facilitate interactions with OME-Zarr. So essentially reading what's in these files and writing things back. This is the baby of Lorenzo, who's here in the picture and who's probably in the audience. Maybe during the Q&A, we can promote him to panelist if he wants to answer some of the questions. If you go to the GitHub that you see here, you will find this landing page along with a lot of the information that I'm going to tell you about here. So again, this is a library that is meant to make it easy for you to interact with these OME-Zarr files programmatically, such that it facilitates the development of analysis method processing methods that operate on this file format. And why did we feel that this was useful? Because as I alluded to earlier, OME-Zarr is very general, which is fantastic because it means that we can put a lot of things in there. It is super useful to put lots of things in an OME-Zarr file, but it also means that it can be a little bit daunting to know how we explore and access and parse this file, right? And that's what I'm trying to get you a sense of here. So of course, in an OME-Zarr file, which is actually a container, right, we have a first layer of information that is the actual image that we store, right? So the actual pixel values. I mentioned earlier that OME-Zarr is this pyramidal file format, right? So we actually don't only store a single version of the image, we store what we call a multi-resolution pyramid, so different resolution level of that image, each of them having their own little pixel grid and positions and so on and so forth. We store metadata. This is the OME part of OME-Zarr, right? That tells us things about what are the axes of the image, what are the channel names, what is the physical pixel size, among many other things. And this is kind of only image information, but we may as well, because that's a container, store other things, right? Typically image-derived information. And you will see that this becomes actually really, really important, in my opinion, if we want to be able to do this like complex, interesting, interoperable processing. But of course, that adds another layer of difficulty, right? Because we can store object labels. We can store groups of objects and say, okay, for each of the level of the multi-resolution pyramid, I can define, I don't know, like segmentation outlines on different groups of pixels in the image that will define objects. Now, I don't have to stop there. For each of these objects, I could also store values, right? Like features that I measure from them, or ROIs that are basically like bounding boxes around them, or other things that I fancy. And these become tables. They can also be put in the OME-Zarr. This is, if I'm not mistaken, not formally part of the specification yet. That's something that we do in ngio because we want to do it for various reasons. Of course, all of this is kind of evolving, but we can store this information in the same container, right? And you may remember a couple of slides ago when I was showing you the sort of zooming in and out on the OME-Zarr through napari, I was showing you this multi-well plate image, right? So we may as well actually have not only one such collection of objects, but multiple, if, for instance, we are looking at this multi-well plate and we actually had multiple acquisition round for each well because this is a multiplexing experiment, right? So for each acquisition round, we may have one such collection, and then this is a single well. So of course we have the whole plate and we can have this type of collection for each of the wells. And now that becomes quite complicated, right? I mean, there is quite a number of different hierarchical levels in this big box that correspond to different things. That, of course, is very structured, and this is what's brilliant about it, is that it gives me everything I need to know about this data in the same box. But that's also a little bit daunting if now I need to do any sort of processing on that. How do I access the different levels, right? And so again, you can do all of this. You don't need ngio for that. But doing all of this is not straightforward. And what ngio does is to make it easier for you. So just to show you, I mean, for those of you this speaks to, hopefully it will make sense. For those of you this doesn't speak to, I will have another slide just after with sort of less programmatic examples. But essentially, what ngio provides are a level of abstraction that allows you to access all of these different hierarchical level in an OME-Zarr in a very, very simple manner. So here, this is sort of showing you, okay, if you start from the highest level, so to say, the sort of big box that contains all the wells, you could say, okay, actually, give me the little box that corresponds to a specific well, B3 here. Okay, let's go. So we get that. Once you get this specific well, you can say, okay, tell me what is the information that I have in this well, in addition to the image, what are the labels and the tables that are present in there. I can simply access it, again, with a simple attribute. And then if I want to actually look at the image data that corresponds to this single well, I can actually also ask simply, give me the image at a given resolution level, and I get that as an array that I can load into Dask or any other format that I prefer, right? So again, you can do all of that without ngio, except this will be many more lines of code, and it has more likelihood to go wrong. So this is just meant to make your life easier, right? And to make our life easier, but then other people benefit from it, hopefully. Now, if you want, I want to see how you can use this concretely to facilitate image processing on OME-Zarr. Lorenzo, in addition to writing this library and making all of these fantastic functions, has also written a very nice and extensive documentation. I invite you to have a look at this. There are examples of how you can use this library to handle OME-Zarr files. I have used it myself as someone who doesn't program so much more anymore. So yeah, I can recommend it. And I hope that this is useful. And again, this is something that is in development that we want to improve and make better in many different ways. So yeah, feedback is welcome. And that's sort of one first step towards allowing easy manipulation of these OME-Zarr files. So now, okay, we have that. What can we do? We can go back to this sort of big ambition of saying, okay, let's try and build processing units that take these OME-Zarr as input, do something on them, and then write back an OME-Zarr along with possible derived information, measurements, whatever, that can then be chained to other sequences of processing units, and that hopefully are modular enough that we can build, whichever analysis workflow that we want. And what I'm going to talk about from now on is a much bigger project that we carry out at the BioVisionCenter. It is called Fractal. It is led by Joël with contributions from many other people. And essentially, the idea is to really kind of make a first step towards this and say, okay, if we restrict ourselves to the Python world for now, can we come up with sort of specification for what would be, an interoperable processing unit that eats, so to say, OME-Zarr files and spits out OME-Zarr files, along with, of course, parameters to say what this processing unit should do and how, and some metadata to say what it has done and how, in a way that this processing unit is defined sufficiently well that it allows us to then chain other such processing unit before and after to make meaningful workflows that are very modular and that can interoperate with each other. And of course, if we manage to define the building block like this, then the nice thing is that it opens up a whole world where we can say, okay, we can build this type of little processing brick to do things like converting files. Okay, that wouldn't start with an OME-Zarr, that would start with another file format, but we can convert things into OME-Zarr. We can do any sort of data wrangling on OME-Zarr through this sort of processing operation. We can do registration, we can do segmentation, we can do measurement. We can do pretty much anything as long as we have this sort of standardized way of entering and going out of the unit that makes it possible to plug it with others that are defined in a similar manner. So how does that work in practice, and how do you build one such unit? Well, if you are doing something in Python that already works on OME-Zarr file, turning it into one of these like fractal building block shouldn't be too hard. If it's already in Python and it's not on OME-Zarr, now with ngio, that should also not be that hard. If it's not Python, maybe you come back to us a bit later. But if you're interested in figuring out, okay, how can you build this type of interoperable processing unit, we do have a template repository with tooling that explains you how to do that. Joel has done some really comprehensive documentation and walkthrough videos. And we can reassure you that this is not something completely crazy that came out of our minds and that only us can do, because we actually have quite a bunch of people now who have adopted this, so to say, and starting to build what we call task packages, so collections of such processing tasks according to the Fractal task specification that essentially all interoperate with each other, right? I didn't put it again here before. I had this little color code of like some of the blocks being segmentation, registration, conversion, and so on. So we have all of these now different collections of processing units that do different things, and that can be essentially taken and assembled at wish to create lots of different things and analysis pipeline that we could not even think about. Now, I mentioned that one of the things that we're interested in doing is thinking about how to make bioimage analysis fair. So the first letter of fair is findable, right? And that's another thing that is interesting about having this sort of specification for what is an interoperable processing unit, is that because we define very well, what are the parameters and the metadata around this task, we can also then essentially list them in a way that is relatively, hopefully, straightforward to explore. This is the collection of task packages that I had here, but now shown in this nice sort of directory that we have, that you can access through the link here, and that allows you to browse, okay, all the things that have been developed according to this specification so far, doing different things on different types of data developed by different people, and so on and so forth. Now, that's great. We have a way to define interoperable processing units. So, let's use that to do image analysis workflows, right? And this is really the core of this project called Fractal that, we are pursuing, is to say, okay, thanks to this specification that is based on the OME-Zarr format, we can show that we can build platforms that can run large-scale microscopy image analysis workflow, and that can be as fair as we possibly can at the moment, I believe. So Fractal, the platform, as opposed to Fractal, the task specification, is a computational platform that allows to combine these tasks together into workflows, and that will essentially provide all the backends to execute the workflows and give feedback on their execution and browse results and so on and so forth. Of course, again, like the specification I just told you about, this is all based on OME-Zarr. The reason why this is possible is because OME-Zarr exists, and because this gives us a very, very solid sort of, starting ground to build upon and to standardize the way that the data is being processed and flows through the path from its input to the final result. And all of this essentially allows us to, is implemented in a way that users can access through web-based interface, such that you don't actually ever need to look into code. So showing you a little bit more how that works. In the backend, Fractal is a platform that is distributed, that is federated. This is a choice that we made primarily for data governance reason. So Fractal is not a service that we offer that we are running it and you can all connect and use it from us. I mean, this is something that needs to be deployed by your local IT specialist or yourself if you feel like you're the local IT specialist. onto your HPC or, I don't know, beefy workstation sitting under your desk or something like that. So of course, this does require a little bit of sysadmin and knowledge to actually set up. But once this is set up, this can be accessed in a completely non-programmatical way through a user interface. And again, the reason why we've made it this way is because the sort of price to pay to have this this nice web-based interface and not have to worry too much about whether your data are uploaded somewhere that they shouldn't be is to have the thing running locally where your compute and where your data are. So from a user perspective, once this thing has been deployed, this is how it looks like. Again, this is just through a web browser. You essentially connect to the platform. You can then run your workflows, design your workflows, explore your data, decide on what portion of your data you run your workflow. I mean, of course, again, this is basically all just leveraging the highly structured and standard way that Zarr stores data. OME-Zarr, sorry, stores data because we use a lot of the metadata here. You can execute, again, a workflow in its entirety, in parts. You have all the usual execution logs you would expect, because what is actually happening whenever you hit run is that jobs are being sent on the HPC, and your image processing is being run on whichever resources that the platform has access to. This allows you to scale very largely, right, and to process very large data sets. And this also allows you to then inspect results sort of by streaming the results also in web-based viewers. That's Vizarr here. I will show more of this in a second. Yeah, so that's kind of a super quick and high-level view of what it looks like to interact with Fractal. As I said before, this is meant to be a federated service, not a centralized one. So we don't run a Fractal instance that all of you can have access to run your own image analysis. You have to deploy your own. However, we do have, as of this morning, a demo server that you can access if you flash this QR code or if you go to this link. You will not be able to do anything there besides looking at the interface and looking at processing results. We don't allow you to run things because, of course, we don't want to allow you to use our very precious HPC credits. But this gives you a sense of the look and feel of the platform and of the kind of interactivity that you can have with it. Like, again, this thing is on since this morning, so I have no idea if you all go to it now, if it crashes or not. I guess Joël is maybe sweating in the background here, but yeah, we'll see. It should work. And if not, we'll discover that soon. Now, yeah, I just want to finish by featuring a little bit what are the really, in my opinion, cool things that the OME-Zarr format allows on top of this interoperable processing, right? And the first thing, which comes back to the first thing I showed about interactive visualization, is that because everything is stored into the OME-Zarr pyramidal file format, we can, again, browse the data super efficiently, but we can also do that in web-based browser. This is here Vizarr, that is developed at the Harvard Medical School, that was led by Trevor Mons. And you can see here how, in the browser, I have one tab that is the Fractal platform, my other tab that is Vizarr, and basically I can just inspect the segmentation results of the data that are on my HPC live, right? And kind of doing this thing of zooming in this terabyte of data or so and having the right resolution load when I need it and looking at whether I think this segmentation is cool or not. This is kind of neat, right? So the capabilities of Vizarr at the moment are fairly limited in the sense that, okay, we can look at segmentation results. We can't do much more, unfortunately. This viewer is under active development, so we hope to be able to do more, as soon as the viewer provides more options for interactivity. If you are not afraid to install something locally, you can also do things in Napari, of course, right? Now, this is just a demonstration of the sort of cool interactive data exploration that we can do on this big OME-Zarr container that come out of Fractal that, again, have all the information bundled together, right? So the original images, associated segmentation labels, associated measurements, and so on and so forth. And that's the work of the fantastic collaborators that we have in Zürich and surroundings that you see here, Max, Fabio, and Adrian, who developed a couple of Napari plugins that essentially allow you to explore the various types of information that is present in this OME-Zarr container in very interactive manner. So the first thing here is basically the same as we did with Vizarr before. It's just saying, okay, because we have the metadata along with the image file, we can say, send me to that specific well of my choice, that specific field of view, and there I actually want to look at the segmentation of the nuclei because, I don't know, I'm super interested in this field of view for some reason. And that's going to load specifically that one bit that I'm interested in. I mean, of course, it may take a little bit of time because you don't need to have this file on your local disk. You can stream it directly from wherever it is stored. And once you have this segmentation, this data loaded and that you're visualizing these segmentations, of course, you also have the associated measurements that go with them, right? So you can do things like saying, okay, actually, color these guys according to a specific feature value. Here, the area, or whatever is going to be next, solidity, yeah? So this kind of gives you just a very qualitative visual feedback, translating the numbers associated with each of these objects into a color. And because you have these numbers associated to these objects, you can also go beyond and do things like classifying objects based on their properties. And that's what you're going to see here now. Because, of course, again, you have to think of this as one big container where we have the original image. For each part of the image, we have associated labels. We have associated labels along with the coordinates of where they are in the image. And for each of these labels, we also have the sort of downstream measurements, right? So we can do things like say, okay, I'm going to click in parts of the image. And whenever I do so, it selects the label, the segmentation mask of the object that I clicked on. And I'm going to annotate these objects according to different classes. Here, red is what are mitotic cells, so cells that are dividing. Blue is going to be interface cells. And because I have measurements associated to these objects, I can then say, from the ones I just annotated, make prediction on everything else. And this is in the background just a random forest algorithm. So the same sort of method you have in Elastic, which I'm sure many of you know. And essentially, this is just leveraging the fact that, again, for this image, we have all the derived information stored at the same place. So we can query all of these different parts of information as we please together. Now, last thing that I want to show with this, again, because we have all of the information together, we can explore it like the video I just showed you, sort of starting from the image, I would say, right? So looking at the image and trying to depict the number back on the image, either through color maps or through classification. But we can also take sort of the path from the other extreme and say, okay, let's start from the data. And this is perhaps more like how image analysis would be approached, like in cytometry or things like that, right? So we can say, okay, let's take our OME-Zarr file, which contains everything, and now load the features that are associated to our object and explore these features, but from a numerical perspective. So we can say, okay, let's plot all of the histogram of the nuclear area for all of the objects in this data set. We can do things like, okay, select, I don't know, a given range of sizes that we care about, or we can do like more gating kind of thing and say, okay, let's look at the scatter plot of, area versus solidity. And because each of these data points is associated to a specific segmentation mask, which is associated to the image, we can also then ask, okay, for each of these data points, what is the corresponding object and what does the image look like? And this then allows us to do things like quality control and figure out that, this sucks. That's absolutely not a nucleus. So let's exclude these guys because we don't want them. And basically say, I only want to analyze these data points for my downstream analysis, right? And when I'm doing this, I mean, yes, I'm selecting the sort of associated features, but I also, because of their link to OME-Zarr, have information about where they are and what type of objects they correspond to. And because of this, I can then do things like exploring, okay, if I do a heat map plot on the plate, on the whole plate for specific features, is there any sort of batch effect? Do I see any sort of like plate-wise weird thing that I wouldn't expect, and so on and so forth? And I can export all of this in whichever format I like, right? So again, it's not that you couldn't do any of this without OME-Zarr. You could absolutely just save every single field of view as a separate image, and then save separate segmentation masks, and then save separate tabular files for all of the measurements, you could do that. And then you could get to something like this if you manage to connect everything to each other. But I would argue that it's probably harder than having it all inside a very nice OME-Zarr container. And that's kind of why we believe in this. Good. So I think, yeah, I'm getting to the end, and it's good because I'm running out of time. What is the most important point that I wanted to make today is that we are developing this thing, but it's actually under active development. This is not a finished product. All of this Fractal task specification, Fractal, the platform, and actually even OME-Zarr, that is not something that I have full control on. All of these things are up in the air. They are being developed actively. They are community efforts. And for Fractal specifically, we made the choice to develop this in the open, to not wait until we have something that we call final to put it out, because we want this to be something that's, we get feedback on and that other people take and perhaps do great things with that we couldn't even think about. So if you flash this QR code, you will land into a page where you have all the different GitHub repositories for these different parts, the tasks, the server, the client, the web client. We have documentation for all of these bits. Yeah, we are delighted to hear what you think about that. If you want to contribute one way or the other, we have monthly meeting for all of the people who are involved in this one way or the other. And yeah, we just hope that it creates nice conversations and nice image analysis, I guess. With that, I'll stop. I want to thank you for your attention. I want to thank wholeheartedly the team at the BioVisionCenter, who has been working very, very hard since we created this thing to do all of these fantastic developments. I did not put my research group on the slide because this was really, really the BioVisionCenter work, but I also want to thank them for their patience because I tend to do too many things at once. And yeah, I'll be happy to take questions if there is still time. Thank you. I'll just stop the screen sharing so that I see people.
Thank you so much, Virginie. There were a few questions which I will forward to you later if you want to post on image.sc. I did not interrupt during the talk because we started a little bit late, but if we still have some time, those of you can stay with us. We can address some of these questions. Is that okay, Beth? Will we still have a little bit beyond the 60 minutes? I believe so. So let's try that. So there were a couple of questions. One, for example, is that if there's an easy way to share light versions of OME-Zarr files when you cannot share the whole data set, but the challenge is it needs to include also annotations and measurements in it.
So I'm not sure sorry, I just found the Q&A box. Okay, so it's essentially asking if we can share, if we could have a version of the OME-Zarr that only has the annotation and measurement, but not the original data. That's the question, yeah?
I believe the full resolution.
That's a good question. I genuinely don't know. Yeah, there are probably people on the call who know, but I don't. So I don't know. This is probably the good thing to ask on Imagency. And I'm sure that, yeah, some of the folks that I see here in the call would be able to give a more inspired answer than what I just gave.
So there was also another talk, and that was one of my questions as well, as to how you see the future of ImageJ and Fiji ecosystem now that we're handling these huge multidimensional data sets. And should we give up hope on ImageJ and just start very quickly training our users on Python and this, or what are your thoughts?
So, that's going to be really my personal opinion. So I'm not claiming at all that this is the absolute truth. But like my feeling is that, so I strongly believe that there will absolutely always be space both for, and that it will be always important to keep both these more like interactive approaches like Fiji, Napari, and so on, kind of next to the more like workflow oriented approaches. Would it be only because, again, I'm saying this from the perspective of a method developer, but I would imagine it's sort of the same from the perspective of a life scientist. Before coming up with an idea of what the workflow would look like, oftentimes you kind of just need to look at the data and play with them, right? And, this is, one of the things that I love about image analysis in general is that it is really, really hard to sort of rationalize about what is it that we want to extract out of an image in terms of information, right? We are just not so good at this as humans. We're good at making sense of images, but not at like rationalizing what is it that makes us make sense of images. And so I think we will always have this thing in biology that is like we acquire image data and we're like, I want to look at this to understand, I don't know, this and that pathway in the embryogenesis of God knows which organism. But I mean, understanding exactly what does that mean, how does that translate into an actual analysis workflow, I think it would always require that we look at the images and figure out, okay, what is it that we may want to measure and in which way? And for this, it will always be useful to have the interactive tools.
Yeah. Still on the topic of interoperability, and there's three questions which sort of intersect, so I'm going to mingle them together. There's a person asking about QPath and interoperability of the OME-Zarr format with QPath. Also, Nextflow, incorporating Nextflow pipelines, for example, with Fractal specifically.
Yeah.
Or the people who are already on the Omero ecosystem, they already have an Omero server. How would they be able to take advantage of that? So I don't know if you can put it all together.
No, super good question. So I mean, I can answer the Nextflow part specifically. So one of the things that we did in one of our past hackathons, I think that was actually the 2024 one, and so it must be, yeah, it is in the paper that we have on that. I can put the link somewhere on ImageSC afterwards. But essentially, we had a group of people trying to think about, okay, if we take processing units defined using this fractal task, can we also use it in other workflow orchestrators like Nextflow, like Galaxy, like actually someone tried, I think, in the napari workflow orchestration, something. So this has been tried, and actually using this sort of building block in Nextflow is totally feasible. With minimal overhead, and that's also kind of what we have in mind, right? The goal is not to say, okay, we build tasks that can only be used in Fractal. Fractal is like one of the platform in which you can use them, and of course, I mean, that's kind of the one we built, so that's how we are thinking about the way we define processing units. But we want this to be general enough that if you want to use it in your favorite orchestrator that is not Fractal, then you do you, right? And conversely, that's we can also use tasks that have been developed elsewhere. So I think this is sort of what we are going towards. Again, all of this project is fairly new, so yeah, everything is in development, but there has been sort of proof of concepts for this, and there is a nice write-up of that in that hackathon report that I can point to. Now for Omero specifically, so yeah, this is a little bit more tricky, and I'm not exactly sure of where we are now. So Omero per se, I mean, this is of course more on the data management side, right? So I guess the question is kind of more, okay, can you connect data in an Omero server and sort of pipe it into Fractal? We have not sorted this out yet. This is one of the ongoing projects that we have together with Jean-Marie and his team. So hopefully at some point this happens. But yeah, we don't have a direct answer for this at this point, I would say.
Yeah, there was also on the QA comment about Vladimir Uhlmann says that it's coming to Fiji soon, so don't abandon hope on Fiji. It's coming soon in the next few months. And there was also another detail from Wei Chen about I also noticed that on the boundaries where you had this large stitched data set, on the boundaries you have, I'm sure you know what we're talking about. So is that a display artifact or what's going on there?
No, so here this is really just because I mean, in this specific data set that I showed, I mean, we didn't do any sort of fancy stitching between the field of view, so we are just processing the field of view independently, and which means that you get this sort of crappy artifacts at the boundary. This can be resolved by doing proper stitching, which we haven't done here. So that's just, yeah, because we didn't do it.
Yes.
But
Yes. All right. Great. I don't know if you have other questions or if I missed one question, please shout out on the Q&A very quickly. Virginie, I will send these questions to you later, and if you don't mind, we'll work on a more elaborated, maybe a little bit, and
no,
linked
absolutely. Very
version on
happy
how to
do
get
that.
more information, and then it'll be posted. Also, keep in mind that this is being recorded, it's going to be edited, and it's going to be on YouTube on the channel as well very soon.
Great. Yeah.
And with that, thank you so much for your time and for everyone's time. My apologies for the glitch on the startup. I'm still new. I'm just starting to help with this, but it was a great talk. Thank you so much, Virginie and everyone. Don't
Thank you
forget
so
the next
much.
session on April.
Yes, thanks a lot, everyone, for coming. And yeah, happy to take questions also as follow-ups over email or ImageSC. Thanks.